Okay, then we switch to the next talk. So Dr. Danny van Hoorn is going from OctoPlus in Leiden, is going to talk about critical considerations in development, scale-up and manufacturing of injectable nanomedicines.
Okay, I first want to thank the organization for allowing me to give this presentation. As you see, I am a program manager at OctoPlus, but also a senior consultant. And I will give you my perspective on what I think that are critical parameters in development and specifically in the manufacturing and of injectable nanomedicines. So specifically I will talk about sRNA products. As this audience will know, there are many companies now developing sRNA type products, but especially because there are a lot of companies doing this, the combination of having good speed, quality, and actually the relation to risk control are very important factors to actually come ahead of the competition and to make sure that your products will be in the market. Although I heard this morning or this afternoon that the regulatory system is still difficult for this type of products. As we all know, sRNA needs to be protected against RNases, but also needs a target delivery system such as a liposome or lipoplex system. And I've heard many others that I've not heard of before. So this is just a schematic overview of how I see that at least the process up to phase 1 could work. Starting with the lab scale, and we see that with many clients, and I've seen it in my past as well. First you need good analytical method development to make sure that you know what you're measuring. So doing some feasibility studies on the development part. Most of the times it's, in our cases, UPLC driven with different detections, and I will come to that a little bit later. So once you have a basis of the analytical method development, you can proceed to start for the formulation development, characterize what you're doing. Let's investigate if what you're developing is actually stable enough at least to proceed. So once you have the couple of ml experiments done for formulation development, you can proceed to a somewhat larger scale, a couple of 100 mLs. If that's successful, the best thing to do is to go to development, and preferably, although not necessary from a regulatory point of view, I would always recommend to start with qualification of your methods, because as you will see later in this presentation, I really think that it's key in the development. So if you have that in place, you could proceed to upscaling of your process to, well, a couple of liters. That is sufficient for tox studies. You need to be really careful if you do that and already know what kind of equipment is suitable, what kind of techniques are suitable for later stages of development, so to ensure that you're not developing a phase 1 product or preclinical product for nothing, because you will not be able to do the same kind of processing or similar kind of processing in later stage of development. So once you have the preclinical material produced what is suitable within development, you should also do a limited ICH stability study that's at least for the length of the tox study itself. So while you're waiting, you don't need to stop, of course, with your development, but what we always do is to have a tech transfer of the methods and the process that you have developed in its smaller scale, to go to the GMP and to do one or more test runs there, see if it's necessary to tweak a little bit the process so that it really fits into the GMP environment. And at the end, the process should be consistent enough to do actual CTM production. So those first three items are most important to proceed to GMP. And if that works, the GMP production itself, at least for the CTM and for phase 1, should not be too difficult. What you need, of course, is good organization also to proceed with your regulatory filing. I will come to that a little bit later. So some examples of what we see with certain clients is that we develop more methods, actually methods that measure the same but use different techniques, just to make sure that later on you can decide which methods work actually best. So on the left hand side you see, I don't know if this works, but on the left hand side you see an ion pairing method for sRNA. The first peak is a duplex, the other one is a singlet. This method is not only suitable for measuring contents but also impurities. And on the right hand side you see a size exclusion chromatogram that's actually measured the same. The peaks are a little bit more closer, suitable for contents but not so much for impurities. So in case of a liposomal products, of course, you need also to be able to detect your lipids and quantify them, and also be able to see the impurities and degradants actually in a stability study. So you need a good resolution on this. On the left-hand side, you see an example of a method that we have developed by UPLC in evaporated light scattering detection. And on the right-hand side, you see the same but different type of lipids. So, in my experience, it's very important that you have good stability indicating assays, and that I think is the most important message of today that I want to give you. Once you have your methods developed, you should also have good quality stressing on those assays, just to see what is happening there, to see if you can have predictive value of what you're doing, also for later stages of development. And I've seen, unfortunately, in the past, some examples that's from companies that did not actually have good methods in place, that was sensitive enough to observe your degradants, and that may lead to later problems later in clinical development, and thereby actually sometimes even in phase 3 that you have to withdraw your product. And one of the things is stability that you did not see. You were thinking that your product was stable, but actually it was not. You only find out later in your efficacy studies. Below you see a picture of an extreme example of stress testing. So the left one is the native formation, the other one is the stress. So we stressed it, in this case, extreme, but just for one picture. So you can see that all was degraded in that respect. What I think it's beside all the technical stuff that has been presented here, I think always that you need effective teams, so dedicated teams that have good information flow, and just to make sure that not only for phase 1 but also for later phases of development, that you're sure that you can go as fast as you possibly can and the authorities, of course, is something that you need to discuss with. But for example, you see formulation development, process development, they will transfer the information to technology transfer and GMP. And you will see that technology transfer and GMP are actually already involved the ATM, that's what we call the preclinical material. So just to make sure that they have a good transition of information, that they understand the process and they understand the critical process parameters. And the same goes for the other departments. So, for instance, analytical method development to QC. One department that I see most of the times forgotten is regulatory affairs. I've done a lot of regulatory affairs myself, so that's why this remark. In my experience, it would be good that regulatory affairs is involved as soon as you possibly can. And I've heard in a couple of last presentations good examples that they were involved soon, because they are at the far end of your filing, and for instance for your IND and IMPDs. But if there was any discussion needed based upon quality, non-clinical or clinical aspects, then they are the ones, together with clinical, for instance, to repair the possible damage. And that should not be necessary. If they can justify because they have been involved before, then things will go faster. And of course, regulatory affairs is not only involved in early stage of development, but also later stage. For instance, for setting a regulatory strategy of your compound and the interaction with the authorities. And I've heard today indeed the need for scientific advice. That's especially for this field is very important to know that the regulators also know what you're doing. So the conclusions, For efficient drug development, a good coordination between all involved departments is needed to go as fast as you possibly can. A production process should be scalable to all clinical phases, and especially true, the critical process parameters should be known early in development, should be tested and tweaked where possible so that you know what you're doing. Sensitive stability indicating assays are a very important factor for successful drug development, and especially for the later stages. And what I just told you, involve regulatory affairs as early in development as possible to ensure to have efficient submissions. That was the last slide. Thank you.
Thank you very much. Have a nice talk. Do we have questions? So, may I ask a question?
Sure.
So, if you envision, let's say, release a liposome system for intravenous use, how much lead time would you think to need up to manufacturing of such a batch?
Manufacturing of phase 1 product, you mean?
Yeah,
or?
phase 1, yeah.
It depends on the complexity of your product, depends on what kind of analytics you have, but it will take, well, let's say about 6 months at least to have that in place.
Okay. Yeah, thank you.
Okay.
You mentioned the importance of release testing. It's also
exactly
referred to by you. What type of testing would you advise for siRNA formulation? Because you know, in the cell, release by the complex is complex.
Absolutely.
and it's competition with other anionic molecules and so on. So, do you have some ideas in this regard? If you want to be relevant for the biological situation, of course you can release it in a chemical way or whatever, but do you have other assays?
I don't have clear examples for that stage, because what you say is true. It's very difficult to have good assays for the biological systems, but that's something we were working on. So that's going to be shared now. Okay.
Any more questions? This is not the case.